Hello everyone, my name is Ziqi, and today I'll be presenting about our poster, Evaluating Deep Learning for Predicting Epigenomic Profiles. Deep learning has been successful at predicting epigenomic profiles from DNA sequences. Most approaches frame this task as a binary classification, relying on p-color to define functional activity. Recently, quantitative models have emerged to directly predict experimental coverage values as a regression task instead. These binary and quantitative models are hard to compare because of divergent ways to predict epigenetic features. As new models continue to emerge with different architectures and training configurations, a major bottleneck is forming due to the lack of ability to fairly assess the novelty of the proposed models and their utility for downstream biological discovery. Here, we introduce a unified evaluation framework and use it to compare various binary and quantitative models for sequence to epigenetic data prediction. This framework provides easy and comprehensive model evaluation, including variant effect predictions and model interpretability for both binary and quantitative models. Importantly, it also allows cross-comparison of models trained in the binary and quantitative regime. In this presentation, I will give a brief overview of some of the provided evaluation methods for performance, variant effect, and interpretability. For all example analysis shown at this talk, we trained models to predict the normalized readout of 15 cell line ataxia experiments and predicted the epigenetic profile for 2K regions. First part is performance evaluation method. As I mentioned, it is important to be able to benchmark new coming models against existing binary models to assess their contribution to this modeling task in a fair way. Our method described here can evaluate models using both binary and quantitative metrics. To convert the binary model output to space of quantitative models, we take the logic value before the final output sigmoid activation. This value is generally considered to represent strengths of signal detected in the sequence. Each logit then represents the predicted strength of signal for 2K chunk, so it's only one value per input sequence. And we compare it with mean experimental coverage in the same 2K sequences. By concatenating these values across the test set chromosome, we can calculate a Pearson's correlation as a quantitative matrix. And to convert quantitative models to binary, we calculate the coverage value in this predicted distribution. We separate them into two distributions, peak and non-peak, according to the ground truth peak calling outputs. With these two distributions, we can now calculate binary metric like AUROC and AUPR. In our analysis, we did see that in case of our models and datasets, quantitative tend to outperform binary models in both matrices, especially when evaluated on whole chromosome, which covers more genomic region with low accessibility signal. Secondly, we want to evaluate models' performance in predicting variant effect. A major downstream application for deep learning models that learn sequence-function relationship is to utilize them to score the functional effects of mutations. High-performing models can inform how their prediction changed relative to wild type when paired with a new mutated sequence. Since we are interested in how mutations can change gene regulation functions, as a ground truth, massively parallel reporter assays, or MPRAs, provide a great validation dataset. Here we use a CAG5 dataset, a publicly available dataset assess 15 promoter regions with saturation mutagenesis MPRA. Promoter sequences were constructed with saturated mutations, which means all three possible mutations on each loci, and inserted into plasmid along with the reporter gene and unique tags. By measuring expression level of tags, we can measure the influence of each mutation on gene expression. Just to note here, the model were not trained on MPRA data at all. They also have never seen the mutated allele in sequence. This is directly transferring it on a new set of experimental data, and here is an example output. Here we plotted the predicted effect size as well as the experimental result in heat maps. Visually, the signals align with the ground truth heat map, which means the model capable to identify important locations for function in the sequence. Also, by measuring the Pearson R correlation between the ground truth and predicted variant effect, we can also benchmark different models in how likely their prediction in calling functional variants are accurate. Here is our comparison result done between binary and quantitative models. We again see that quantitative models, on top of better prediction performance, also show better variant effect prediction capabilities. We also notice that robust predictions generated by averaging prediction across random translations yield better prediction performance. If you're interested, you can find more analysis detail in our paper. Lastly, we incorporated global importance analysis, or GIA, into our framework. While attribution maps can help to identify and footprint possible motifs, they cannot quantify the importance of motifs beyond individual nucleotide. GIA is an interpretability approach that enables direct testing of hypotheses gained from attribution analysis. GIA computes the effect size, or global importance score, of hypothesis patterns that are embedded within a population of background sequences, which the other positions are effectively randomized. This approach essentially marginalizes out any confounding patterns within an individual sequence, revealing the global importance of only the embedded pattern on model prediction. Here, we show an example comparison of two models trained on our same ATAC-seq dataset. We can see that they both identify the AP1 motif as important for the task. However, they show different sensitivity to motif interactions. The model above, which is a quantitative model, identified two AP1 motifs in the same sequence, is actually more important than twofold of a single AP1 motif. This suggests that this model has learned interaction or cooperativity between motifs, while the one below, which is the same model structure but trained on a binary task, did not identify this interaction. Therefore, GIA can be used as a method to identify as well as compare motif syntax learned by different deep learning models. Due to time limit, we only included an anecdotally chosen comparison here. We have also used GIA to explore other features, including importance of flanking nucleotides and distant dependence between motifs. We also compared the filter interpretability on different architectures. Check our paper for a more comprehensive and systematic comparison across different model design. Thanks for listening to the talk. If you're interested, check our preprint on bioRxiv. Thank you.